All right, so for chapter 10 here, part 2 for photosynthesis, we're just going to briefly talk about the electromagnetic spectrum and light and how this is involved in helping us perform photosynthesis. So, of course, the electromagnetic spectrum is the entire kind of gamut of radiation that's given off, all the way from, the radio waves to gamma rays. So it spans the entire spectrum, and there's only a small portion of that we can see. And of course, you probably learned in junior high or something Roy G. Biv for the colors: red, orange, yellow, green, blue, violet. And these this end here, the red has the longest wavelength, and then violet, of course, has the shortest. On our way down from, on this end here, we'd have radio waves. And then down on this end here, we'd have the gamma rays, okay? So it makes the entire span. So if we look at it, this is kind of reversed now from what we just said with Roy G. Biv, but here's our gamma rays over here and all the way to the X-rays. So when you look at it, this is the red end, okay? And then this is the violet end. So it would be Roy G. Biv kind of going in that direction. So it doesn't really matter which way you write it, longest to shortest, shortest to longest, just as long as you know that red has the longest wavelength and that the violet has the shortest wavelength, okay? So now, when light hits an object, there's three basic things that can happen. You can either reflect it, you can absorb it, or you can transmit it, okay? And when white light or some other color hits the light, what you're seeing is the light which is being reflected. So in other words, if you have a white shirt on, it's white because all colors are being reflected. If it's red, then red is the only one being reflected. The other colors are being absorbed. And for our purposes, for instance, with chlorophyll, leaves are green because they're reflecting the green light. Chlorophyll doesn't absorb green light very well. It reflects it and absorbs most of the other colors, and that's what it's going to use to kind of drive photosynthesis, okay? So we said earlier that chlorophyll A was the main photosynthetic pigment. Chlorophyll B is another type, and then you have some carotenoids and some xanthophylls, if you remember from the labs that we were doing, and if we haven't done them yet, we definitely will. But chlorophyll A basically is going to absorb the violet, blue, and the red light the best. So you can see right up here, here's your chlorophyll A, and if you follow the kind of the brighter green line right here, you can see the different types of colors that it's going to absorb. The chlorophyll B is kind of more of a drab greenish color, army green, and this absorbs violet, blue, and red, orange. And then the carotenoids, these are going to absorb violet light very well, and then some blue-green light. So you can see the chlorophyll B, that's this drab color, and then the carotenoids, these are the yellow colors that you're seeing as they go through there, okay? So chlorophyll A, again, that's the main photosynthetic pigment we said, and that's what the main driver of photosynthesis is. And chlorophyll B and the carotenoids, these are referred to as accessory pigments. So what they do is basically broaden the spectrum of light that can be absorbed and used by the plant. So the useful photosynthetic pigment is basically expanded by accessory pigments, things that are, just kind of helping make more efficient use of the light, okay? And then one of the other things that they do is they serve a photoprotective kind of a purpose. And what they're doing is they're absorbing some of the light energy and helping dissipate this as heat that would otherwise damage the plant. So by broadening that spectrum, they're enabling the plant to survive in very, very hot, warm, sunny conditions that occasionally occur in temperate regions and such. Okay, so as far as experimenting with light's concerned, the action spectrum of kind of photosynthesis was, I guess, paired up or matched well with the absorptive ability of the chloroplast. So looking for the effective colors versus the ineffective colors. And if you recall, one of the main byproducts of photosynthesis is oxygen gas. That action spectrum or the amount of oxygen gas that's being produced can kind of be overlain with where the colors of light are kind of being optimally absorbed, okay? So if you think back just real quickly to when we were using our spectrophotometers in class, what we had, if you turned it on, you can remember it beeps and then it kind of makes some noises, and what it's doing here is it's kind of calibrating this prism. So it's kind of moving this thing back and forth here from the light source that's on, and what it's doing is it's optimizing what color of light we want to go through the little slot that we have in this machine. So when you set it, for instance, to 520 nanometers or whatever it happens to be, it's going to allow that particular color to pass through. Okay, so in other words, we said that the chlorophyll solution does not absorb green light very well. Okay, so if we set our prism up to drive us some green light through here, most of it's going to pass through to the other side of this detector in our photosynthesis, and it's going to give us a reading here, which is going to be very close to, for instance, 100% transmittance. If we were looking at absorbency, then it would be down here. It would be a very low absorbency. So it's going to transmit a lot of green light. It's going to absorb very little of the green light. Okay, so that's how they know that this is that chlorophyll doesn't absorb green light very well, just from the experimentation and some of the other things with physics that we know about. All right, so here again, this is our action or absorption spectrum. So we can see chlorophyll A, chlorophyll B, and the carotenoids, some of the things that we went through here, kind of discussing the different wavelengths that they absorb at. And then we can kind of look at the action spectrum. So this is the kind of rate of oxygen release. Okay, so if we're looking at this, we can see here the oxygen release is high, here it's kind of low, and then here it's kind of high again. And if we kind of compare these things too, you can notice that the points where they're the greatest are where the majority of the light is being absorbed. Okay, right here and right there. And then here you can see it's very low because there's not much being absorbed. Okay, and that leads us to kind of an experiment by Theodore Engelmann, who kind of famously set up this very difficult experiment. And what he was doing was he was experimenting with some spirogyra, and that's a type of algae. Okay, so here's the spirogyra right here. And algae, of course, have chloroplasts, they're plants, they photosynthesize, etc. Well, he also took, and on a slide, what he did was he put some aerophilic bacteria or aerobic bacteria that really liked oxygen production. And then he laid this, we can look at this here, and this is like his microscope slide, okay? So this all the way along here is his microscope slide. And then what he did was he set up a prism that would just put different colors of light all the way along this particular slide. So you basically had the rainbow of light kind of over the slide, and on that slide now he laid this spirogyra. Okay, so now he's got algae that's lying all along there, and of course algae photosynthesize, and they absorb light optimally in these conditions, or in these particular spots that we'd been talking about. So that's where we would expect to see the most oxygen produced, and hence you can see all these little speckles here, the most bacteria kind of congregating in these areas. And of course in the green area, it doesn't absorb very much light at all. It's reflecting most of that light. Therefore, there's very little photosynthesis and very few bacteria congregating in those particular areas. So what he found was that these bacteria were congregating in the areas of the blue and the violet light, the red and the orange light, because that's where the most oxygen was being produced. And that's kind of how they got this action spectrum overlain with this absorption spectrum. Okay, so that takes care of photosynthesis part 2. We'll be back to talk about the light dependent and light independent reactions in part 3. So we'll see you soon.